Our last section in chapter 3 is basically just kind of a wrap up of some of the processes that we were seeing in meiosis. And then the last little bit is a specific discussion on production of sperm versus eggs. So here we have a list of the sources of genetic variation in meiosis. If you want to pause and write them down, go ahead. They should all be in your notes already. Here we have another diagram of crossing over. I like this diagram because it shows step by step what happens with the alleles on our chromosome, which is a really great depiction of how our chromosomes are exchanging information and what that means at the end, because we are about to move into Mendelian genetics, where we're going to talk about those alleles and how the alleles and their associated traits get passed from parent to offspring. I also like that this diagram shows you the new types of chromosomes that we have. So if we think of these chromosomes as a representation of what would originally be this individual's parents. So what we have here are parents, and then the individual who's producing gametes. And then those gametes are what are going to produce the child. So we kind of see the link here between 3 generations of individuals. So this cell here would be parent, so think your parents. And then when your cells undergo the process of meiosis, they are undergoing crossing over. And so when your gametes are produced, this is what's happening. And any child that you have as a result from your gametes are actually going to be combinations of their grandparents' chromosomes. So yes, they are combinations of your chromosomes, but your chromosomes are a reflection of your parents, and it is actually your parents' chromosomes that are doing the crossing over here, which is a little bit crazy to think about. So during this process, let's assume that this is mom's original chromosome and this is dad's original chromosome, and then think about this is the individual producing the gametes, so their parents' chromosomes. So if they exchange on their center sister chromatid, so we're going to go through DNA synthesis, exchange at the center here, so we're going to exchange here and here. And the result is that the outside chromosome is exactly the same as the original maternal chromosome. The inside one is now a combo. These 2 chromosomes in the middle are a combo of the alleles from the parent chromosomes. So then when they are separated by the end, we have those 2 chromosomes that are identical to the original parent because they were not participating in this crossing over event. But the 2 that were in the center are now brand new chromosomes that are combos of alleles from the parents. So we get these new unique chromosomes in the center here that are new combos of alleles. These are unique and new combos that weren't together before. Here we've got a diagram of random alignment, random assortment, independent assortment, whatever you want to call it. Just make sure you know the different names. And I would say stick with whichever one you want to call it. I like this image because it's a little bit more complicated. I'm not going to ask you to draw out something like this, but it does show you, again, from the parent chromosome, and then when the individual is producing their gametes, we get these potential combinations. So in column B here, it shows all the different ways, well, not all of them, several of the different ways that these chromosomes could line up. There are actually 2 to the 3 different ways that these chromosomes could line up. So we don't have all of them here, but that's okay. This is plenty big. So you can see that some of them are flip-flopped, lined up a little bit differently, and they're going to give you different combinations. And then we have the result here. If option 1 happens, these are your outcomes. Option 2, these are your outcomes. So you can see no matter which way they line up, they're all going to be a little bit different. And then also keep in mind that we have crossing over as well. So even if we don't get any new rotations of our chromosomes, we are still going to have chromosomes that are a little bit different here because some of these are going to be the result of that crossing over event. So we've got that double impact of crossing over plus random alignment. So remember, crossing over is going to build these chromosomes that are new combinations of alleles, and then random alignment is going to build these cells that are new combinations of entire chromosomes. Now we're going to shift gears a little bit and talk about what happens when we actually separate our chromosomes. Before I get into all of the details on the slide, I want to point something out, and that is that our textbook and I draw our chromosomes like this, kind of in an X shape, where each side is a sister chromatid. But in reality, they're not just flailing arms all over the place. What actually happens is that our chromosomes remain straight, and they remain connected all along their edges, kind of like a zipper. And then when nope, I'm not going to go there yet. The way that this is held together is essentially a ring of proteins that sort of strap these guys together, and then a little collection of proteins that hold them right down the center. And we have a couple of proteins that are involved in this process. So the first is cohesin. Cohesin is a ring-shaped protein that is going to hold these chromatids together. And this is important in keeping those chromosomes together so that they separate correctly. Because if they separate incorrectly, we end up with altered chromosome numbers. So for example, Down syndrome. So we want to keep those chromosomes organized. We want to make sure that they are moved correctly and then they're separated correctly once we get to the metaphase plate and pull them apart. So there are 2 points when cohesin is important. One is to hold the tetrads together, and then also to hold the sister chromatids together. And cohesin is going to work to hold them together until they are ready to separate at their separate points in meiosis 1. So there's a second protein here called shigoshin. Now shigoshin basically keeps cohesin together. So we've got cohesin and we've got shigoshin. I would suggest flashcards at this point. So cohesin is held together by shigoshin. And during meiosis 1, when our homologous chromosomes separate, Shigoshin breaks down, which allows cohesin to break down at the right time, and homologous chromosomes separate. Then when we get to meiosis 2 and our sister chromatids separate, more Shigoshin breaks down, releasing the cohesin that's holding the sister chromatids together. So here we've got our sister chromatids during mitosis. So just to kind of explain what's happening here, this is our double-stranded DNA. And it's not drawn here just for simplicity's sake. So that's our double-stranded DNA. So this is one of our chromosomes, and this is our second to form the sister chromatid. So this image here is this guy. Each one of these gray bars is one of our chromatids. Here we've got the cohesin ring, basically holding the chromosomes together. So we see two different depictions here. You've got this ring holding your sister chromatids together in the center. All right, so during metaphase, our chromosomes are all lined up. When they are ready to separate for anaphase, cohesin breaks down. So there's our cohesin, we've got it breaking apart, and then our chromosomes can split apart and pull towards either pole in the cell. The same process happens during meiosis. It just happens at two different places. So in this image here, we've got our sister chromatids, and we've got our homologous chromosomes. Now you can see cohesin is holding our sister chromatids together. And then we've got our, when our chromosomes are ready to separate, our cohesin is going to break down. You can actually see here, we've got that crossing over. I'm just going to write that up here. There's our crossing over. Just a little quick diagram to remind you that it's happening. We've got our chromosomes connected at the kinetochore. All right, so we've got cohesin along the chromosomes to hold the synapsis, or this says chiasmata, that's another term for synapsis. And then our chromosome cohesin starts to break down so that during anaphase 1, our sister chromatids remain intact, but our homologous chromosomes are stuck together. So cohesin starts to break down, and then they're going to separate. When we get to anaphase 2, our second round of cohesin breakdown is going to happen. Remember that Shigoshin protein is going to hold our cohesin together to keep those sister chromatids together while our homologous chromosomes separate. So we've got that Shigoshin regulating what is holding everything together. So finally, once all of our cohesin is broken down, then our chromatids are going to separate.